All right. All right, welcome back. Do we have singing in the background? I could record this if we want to.
This is just what we
The-
do during lab.
Okay. Music makes things go better. I guess I'll go for that. All right, so the two things that we want to talk about, folding funnels and clinical significance, okay? And actually, I'm showing the screen because I'm going to take my friendly unfolding protein here, and I'm going to show you the idea. So the TM problem with collagen shows you that if you can measure what percentage of a protein is folded or unfolded, you can get a TM, and that will give you a single number that tells you about its stability. So in a sense, as you increase denaturant or increase temperature, you start off with it folded, 0% unfolded, and then you have a narrow range where you end up with partially unfolded signal, and then you end up with a long range where you get completely 100% unfolded signal. This is a cooperative protein. It has a very sharp and steep rise. But remember, apomyoglobin is a broken protein, will tend to have less cooperativity. They've looked at to see, okay, what's going on at the 50% point? Because there's two possibilities. Either you can have there's two ways to get to 50% unfolded. You can have 50% of your sample 100% unfolded, like this, or you can have 100% of your sample 50% unfolded. You see how that works? All you're measuring is a signal, so you don't know from the signal which way it is. Is it 100% like this, or 50 like this, 50 like that? When they've looked at it, especially for the proteins that you can look at easily, you can see that there's actually a mixture of, it tends to be 50% like this and 50% like that. In other words, that means in the middle, you have 50% like this, 50% like that, and very few things in the middle of that. And that tends to go along with a reversible folding with a sharp transition. So they call this cooperativity. In other words, if you start to disrupt the hydrophobic core, the rest of the protein falls apart. It's like a Jenga thing. If you take apart one brick, then the whole thing falls down. It's on the verge of stability. And once you start to trip over that edge, then basically the rest of that particular molecule is going to be disrupted. This is what cooperativity looks like. It means that the ideal protein will not have many structural intermediate states that you can capture. It won't have many stable intermediates, because that's what 50% folded, a molecule that was half folded would be half unfolded. That would be a stable intermediate. Like that I state that we showed on the previous one. That would be half folded. Things like ribonuclease will be two-state proteins that will be well behaved in solution. And if they're like this, it also makes sense that these will be more reversible, because it'll be easier to go back and forth. You won't have these half-formed proteins that will be able to aggregate. And so protein folders tended to study these. Now, whether these are representative of proteins as a whole, there's definitely more of a spectrum in real life. But I want you to know that protein folding can be very cooperative. It can be, once you add enough temperature, heat temperature, to start to make the core fall apart, the whole thing continues to fall apart. Lose one interaction, and you lose all 10. It's that kind of thing. So ideally, protein folding is an all-or-nothing process, and most protein folding studies have been done on ideal two-state cooperative proteins. Realize that all the proteins still have intermediate states. It's just they're not stable. You only see what's stable when you determine a structure or you put it in a tube, unless you have very advanced equipment, or unless you're doing it in a computer where you can stop it at any time. So it's hard to observe or stabilize them. You have to get from point A to point B by going through an intermediate state, right? It's just hard to catch it, just like the transition state is hard to catch. So it's the same kind of thing. Does it even really exist if you can't isolate it? That's a philosophical question. Realize that the unfolded state also, so the intermediate state is there even if you can't catch it. It's just a question, you have unstable intermediates for a ribonuclease like a two-state protein. And even in the unfolded state, it's not going to be completely chaotic. There are usually little flickers of structure in the unfolded state where it's kind of doing like this in a really complicated kind of way. So remember that the unfolded state is not a pure random chain. It's just unfolded. It's unstructured, and it's dynamic. It's moving around more, but it still could be moving around in a reasonable way. You still can study the structure of the unfolded state. So the important thing is protein folding is cumulative, but it's also extremely fast. And in the cases of highly cooperative proteins, all of the interactions depend on each other. If you take one out, all the rest fall apart. So how does this relate to, how do we think about this? I want you to use the idea of a folding funnel. This is, there's actually, I know the scientist who came up with this, and it's really taken off because it's a really good way to sort of imagine what protein folding will look like. The advantage it has, it's sort of thinking of it in 3D with the folded state all the way down here and the unfolded state at the top of the funnel. So when you have a protein folding, and there's like some, I don't think you can hear it on the recording, but there's like some chainsaw going on out there or something. It's just in time for Halloween. So, but you have a multi-dimensional landscape. The idea is the protein originally is in its unfolded state, and it's rolling around. And then you either decrease temperature, maybe it's in its unfolded state because you have a lot of denaturant or a high temperature. But it's rolling around here, and it's unable to access its folded state. But then you lower the temperature enough so eventually it's going to fold. And so it's rolling around on the top here, and then it sort of rolls down and compacts a little bit. So if it has a stable intermediate, that's shown right here. Stable intermediate, it will sort of half form and sort of roll around in this. But after it's rolling around in this part, then it will be able to just get over this little transition state and fall all the way down, find its folded state. And once it's found its folded state, it's happy. It's at low energy. The cool thing about this representation, energy is on the it's on the y-axis. The y-axis is going up and down. And sort of the x-axis, the xz-axis, the sort of flat surface that's rolling around, the width of the funnel is its entropy. Remember that when you're starting to roll down this state, you're still in a high entropy state, but as you go down, the energy gets more stable and the entropy goes down. This captures this aspect of protein folding, the unavoidable entropy loss that you have to go through to form the stable structure down here. So according to this, entropy decreases during folding. Energy becomes more stable. It becomes more negative, but we're going to be chemists. But entropy becomes, energy becomes more stable as entropy decreases. The unstable intermediates are sort of everywhere, but right here, this is a more stable intermediate. But this is a, and the unstable intermediates will be the ones with shallower dips. The dips are intermediates, and the question is, do you see them, or does it roll right past them and into the final conformation? The deeper they are, the harder it is to roll right past them. The most stable conformation is right here. The thermodynamic minimum is where you have the least energy, the lowest energy, the most negative energy. That's why I don't want to say least, because it's most negative is what we're talking about here. So you can actually make a protein folding funnel, and again, I wish I could use a 3D printer. I could make one of these and pass it around, but at least you can see what it looks like. This is a protein with a lot of stable intermediates, because you see it has these stalactites like pointing down. And you imagine rolling down on this, it's pretty easy to get caught in one of these things. So it's pretty easy to have a stable intermediate, but it's still just an intermediate. If you're able to get out of it, you can roll all the way down and you can fold at the very global minimum. So not all proteins are going to have the same folding funnel. In fact, it's very helpful to look at different protein funnels. For example, like a nice stable ordered protein, these are actual folding funnels, by the way. On the left is an ordered protein funnel, and it's pretty smooth. You have like one stable intermediate over here, where it can sort of get stuck, maybe if it rolls down that side of the funnel. But there are some proteins that are more disordered. And this is a protein that's sort of going like this. It doesn't even fold very much at all. It has like 4 wells that it kind of floats around between, 4 different twists. So this is a very broad and shallow folding funnel, more disordered. So order goes with the smoothness and depth of the funnel. And so this is another thing that helps you think about how to think about funnels. Here's 4 examples of funnels. This one right here has many paths to folding and few intermediates. It's very smooth. This might be like ribonuclease A. So a smooth, deep funnel will be very cooperative as well, because the intermediates are what will trap you in and make your protein folding curve actually be more shallow. Intermediates will do that to you. This one right here is sort of an idealized one where you have very few paths to folding. It's hard for it to find the folding. But once it finds the crucial, weird configuration, everything else snaps into place. So this one will actually be also a very cooperative protein. It's either all or nothing. There's not much intermediate there. This one has one necessary intermediate. No matter what the structure of the unfolded state, it has to go through this dip, this necessary intermediate, before it gets to the final structure. And then this one is what reality usually looks like, okay? For this, there will be lots of different intermediates depending on the way the unfolded state is twisted. Which way it's twisted will encounter different intermediates, but at the end of the day, you have one native state at the bottom, and that's a nicely folded state the global energy minimum. This is very helpful because this is, first of all, it's cytochrome C, which you'll work with this week in lab. But it also shows you very clearly what the structures of the different points of the funnel are. It has these structures, and as you go, you start from the unfolded blue protein, and you see that it's very spread out. It has higher entropy, and so but it has worse energy because it's not making good interactions. As it moves down, it starts to form different structure, like the green structure forms, then the yellow, then the red. Each step, that area of the protein will compact itself, okay? And so each step, the entropy goes down, but your energy is getting better. That means you're forming lots of good, stable interactions, and you're forming structure that compensates for the loss of energy, the loss of entropy in the form of energy known as entropy, right? That's what it means when it says that the funnel's getting narrower. The structure itself is getting narrower. And so you're forming good interactions, and at the end of it, these 4 interactions form in this order to form native cytochrome C at the very bottom of the funnel. The least entropy, but also the most stable energy, because it's forming all these good interactions to compensate for the entropy it lost. The folding funnel really helps to deal with managing, thinking about entropy and energy at the same time. That's the real advance it is. And that's the conceptual thing that I sort of want you to get from this lecture. It's very hard, and that's why they do it. So there's a couple of papers that just came out just to finish up talking about folding funnels before we move on to clinical relevance. First of all, why is the brain better? Why does fold-it work so well? And David Baker put out a paper about this just in the past year, where he studied how people solve protein structures and why it's better. The answer is you will take risks that a computer will not. It's actually, I've heard it's also the way to beat a computer in chess. You take risks that the computer will not, that don't make any sense to it. And it could be even an irrational risk if it works out okay, or you could lose badly. So, but if you take bigger risks, you get yourselves out of the ruts in the funnel. You get yourselves out of the stable intermediates. And there's actually a figure that shows this. The important thing about looking at this figure, the gray lines are the computer reasoning, the computer running the folding program. The different colors are different humans that are running the folding program. And so notice that this is relative energy, and you're trying to design a sequence to fit a structure. So what you're doing is you're putting in different mutations, and you're trying to get a backbone structure that you've determined already that you want. One of these structures, one of these 6 structures shown up here. And so the computer and the humans are trying to do it. The computer is actually more efficient, but you see right here, it's got the energy down, and the gray lines all go quickly down, and then they stay down. Because the computer's like, I've got good energy. Why should I break this interaction? And you try to program that in, but it's just hard to get the computer to take a risk, break a good interaction to try to form a better interaction. Notice that the humans are not as efficient as the computer, but once they get down here, they will take risks. They'll do mutations that basically increase the relative energy. They're not happy with the relative energy being low. They will continue to take risks that are bad, small bad steps, hoping that it will eventually allow you to take a better step forward out of that. It's shown in another way for this. This is showing the design, and it's where you make a decision and how many things you try based on that decision. It's what they call branch points. And so you start with this particular structure, and I believe this was a cooperative one where like the red player would do a design, and then the blue player and green player would go in. But you see you have the red player trying a design, and they're only able to try like 20 different iterations of that design. But eventually they find one they like, and they do 40 different iterations of that one. You know, they have fewer branch points, or I mean they have fewer structures that they try. But eventually they actually are able to, they try fewer things, but they make more decisions, more branching decisions, more risk-taking places. The computer's tree looks like this. Basically, it's 5,000 wide. It doesn't even fit on the page. But there's only 2 branch points. Like it picks this one and then makes 10,000 of those. This is the brute force computer computational way to do it. This is human reasoning. And you see how it's actually getting lower. So humans can't do 10,000 different structures. It can't try out 10,000 different twists. But humans can be more risky. And so they can find the hidden pathway through the ruts in the folding funnel, because they can sort of push their structure out of the little rut to get it over that little energy thing where you can roll down and you can get to the bottom of the well. It's kind of conceptual, not really that much on the test question, but I want to say, what do humans have that computers don't? Here you go. You got this. Okay.
Would it be therefore most effective to have a computer just make a bunch of them and then give us humans like the most possible beneficial ones and then have us screw around with them?
Yeah, but the thing is, the sequence space is so large, the computer, anytime you are giving a human, you know, you end up with, a human can't go through 10,000 structures.
Yeah, but that was, like a computer could just like select the best ones and then have us go around with
Yeah, but the best ones are all within this sort of relative range, and it's still stuck in a local minimum. It's just the nature, the conformational space is so huge that even a computer with 10,000 samples is only getting the little corner of that space. You've got to take big risks to break yourself out of that corner. But there's some kind of combination, some kind of thinking that does combine the two. But it's trickier than it looks. Yeah. So the thing about the protein funnel, it actually also explains why there's protein folding disease. And that's the topic I want to end on. The protein folding funnel, like I said, most proteins, thankfully, end up in the native state. But there's a lot of proteins that have another state that has a low energy conformation to it. Hopefully this is hard to get to, but let's say that you have aggregates. Technically, stable aggregates are a stable state that you've sort of fallen into. You could show them on a folding funnel as being like a red place down here that you don't want to go to. You want to move the protein toward this shape. You don't want to move it toward that shape. And if it gets to this shape, you can't reverse it. Like we said, precipitation is often irreversible because the energy well is so deep, you can't climb out of it again. Sometimes you even have something like this, where you have a really bad repeating conformation that is really stable. And if you get into this well, you start to make a stable structure that will repeat, and it will force other proteins to possibly find that same structure. That's when you have the possibility of making not just an amorphous aggregate, but a structured fibril, and that's called amyloid. So amyloid diseases have to do with the protein folding. So here's an example of the different kinds of things that you can see for a green fluorescent protein. They studied its folding funnel, and they found out, for example, it's unfolded is up here, and it moves down to the lower right to form the folded native state. But it's got three different stable intermediates that it could form that are all twisted in weird ways. And it can actually take any of these paths, but there's a certain percentage that will take it, and there's a certain stability it will have when it gets there. This is another example of a folding funnel to understand this very complex interaction. But notice that these three stable intermediates, they sort of trap the protein. They are more stable than the surrounding conformations. So if the protein gets down in here, it will take some twisting and some effort to get it out of there, some energy. If this well ends up being too deep, it becomes a kinetic trap, is what they call it. It traps the protein in that untwisted conformation. And check out this one right here, right in the middle. It has this blue beta structure that's sort of sticking out. What if this meets another blue beta structure and they start to hydrogen bond? You start to have a repeating structure that is open at both ends because each end can hydrogen bond to another beta structure. Even a nice little protein like green fluorescent protein has the potential to form repeating beta structure, and that has the potential to form an amyloid fibril. So that's why you get pictures like this. You have, remember that I believe I showed this way back in the homeostasis lecture. You have proteostasis, you have all these different forms of the protein, and you have to get from unfolded to folded, but you have all these different things going around that are changing the possibilities. If you have a high concentration of unfolded protein, you have the possibility for aggregation over here, which moves you to the right side of the screen and ends up forming amorphous aggregates, oligomers, or worst, amyloid fibrils. The cell does have mechanisms to find these and chop these up, but that's molecular machinery, that's cell biology. What we're talking about is protein folding that causes the problem in the first state. This is even ribonuclease A. So I've been talking about what a great protein ribonuclease A is, right? Even it can form some fibrils accidentally. This is an example of some conditions they found where the ribonuclease A sort of unfolded. It had beta structure sort of flapping around, and you had a high concentration of it, so it started to stack up. And you see right here in the middle, you see the beta structure. And the beta structure has hydrogen bonding potential on both sides of it. So all you have to do is have a high concentration of the protein where this starts to stack up, and you start to get an amyloid fibril in the middle of solution, even from the best behaved model citizen type protein. Okay? So this potential is always there. Because beta structure is open at each end, it has a propensity to be able to misfold. This is why protein folding can save your life, because it can actually treat these diseases. If they are problems with misfolding, maybe we can come up with some way to understand that structurally and then stop it from happening. So it's implicated in the following diseases. Actually, you tell me, amyloid diseases. Do you know of anyone's? What's that? Alzheimer's is a big one. And by the way, it's the protein that we made in class is Alzheimer's protein. But there's a lot of other ones too. So Alzheimer's is the big one. Any other ones?
Are there blood diseases, like blood clotting perhaps?
There's sickle cell anemia, which is sort of a different kind of thing. It's not based on the protein folding. The thing about the blood is if you form amyloid, you can get rid of it pretty easily. But there is a way that it forms from hemoglobin, but we'll talk about that in the next chapter. So that is certainly a thing. It's a bigger problem when you form the amyloid in a place like the brain. So a lot of these are brain diseases because it's harder to clear out of there. Parkinson's is one of them. Oh, and another one?
I forget how you say it, but like Jacob, and it's like that.
Yeah, those are prion diseases, and that's a whole topic. It's sort of a topic in itself where the other protein actively catalyzes the misfolding of other proteins. We'll just mention those here. They're a slightly different category, but they're very cool. I just don't have time to go into them. But yeah, the prion diseases are misfolding diseases. Cystic fibrosis technically is a misfolding disease. It doesn't form amyloids, but a mutant protein misfolds, and you can try to solve it by studying how the misfolding happens. If you've ever seen Unbreakable, Samuel L. Jackson has osteogenesis imperfecta, which is misfolding of collagen. That goes along with the foundations problem that we have. And therefore, if he just falls down, his bones will break because he has brittle bones. Alzheimer's, misfolding produces beta amyloid plaques. And so that's what we'll talk about. And prion diseases. These are all sort of different categories. I'm going to focus on the Alzheimer's category. Because when you have amyloid in brain cells start to die if they can't clear it. And what you end up with, this is one thing that happens in prions, another brain-based disease. It's called spongiform degeneration. So you don't really have to be a doctor to see the root word of spongiform, right? So spongiform means spongy brain. And what happens is you end up with these holes in the brain, and this is caused by the prions misfolding. It's a little more subtle. You don't get big holes in the brain with Alzheimer's. Instead, you have these plaques. But I want to show that you have the plaques. There's multiple kinds of Alzheimer's. So here's one that shows, in fact, there's multiple proteins within Alzheimer's that form deposits. Amyloid beta are the main deposits in Alzheimer's disease, and that's shown in orange at the top here. You get those in these regions of the brain. They look like that on the left. There's also tau proteins that form inclusions in Alzheimer's. And so it's a question of which of these is more important. I'm going to talk mostly about the first one, but realize there are even other proteins in Alzheimer's. Parkinson's disease is in red. That forms inclusions of other proteins in these areas of the brain. And finally, amyotrophic lateral sclerosis. There's a more common name for this. Anyone want to guess what it is? ALS. Exactly. This is Lou Gehrig's disease. And it forms in different regions of the brain from a different protein. But here we have 4 different proteins, 3 different diseases, 4 different proteins, different areas of the brain, same basic problem. You have amyloid misfolded fibrils. And that's because when you have a repeated structure, especially beta structure, you have the propensity for the beta structure to self-associate. Now, if the protein folds itself really fast, you don't have that exposed beta structure usually. The beta structure is hidden on the inside of the protein. But if you have a stable intermediate, or here it's called a molten globule, if you have open beta sheets, those open beta sheets can self-associate and form a fibril. And this actually also takes place in Huntington's disease and even some forms of type 2 diabetes. This is bad news. So Alzheimer's, this is from your book, and this is low resolution. But Alzheimer's forms from a 40 or 42 residue fragment of a protein. And it turns out that this fragment self-associates. The fragment starts off as alpha helical, but then the protease is trying to clean it out and clipping it up like that. But if the job isn't completed and you have a lot of this peptide around, it will stretch out to form beta conformation. And once you have the hydrogen bonds forming up and down, you also end up with repeated phenylalanines. And you know from your paper, phenylalanines and phenylalanines will interact well. And so, in a sense, the side chains are helping this fibril form with the repeated phenylalanine structure. They actually found this with X-ray crystallography, and I want to show you this because remember that X-ray crystallography tells you about the spacings between structural elements, the spacings between atoms. So they took the fibrils, and they actually ran the X-rays through them, and they got this data right here. They got really smeary data because the fibrils are not really well ordered. They're not crystalline. They're not the good topics. But they were able to squeeze out from certain fibrils in certain forms, they were able to squeeze out a refraction pattern that looked like this. And you actually have a 10 angstrom reflection and a 4.8 angstrom reflection from that, they were able to figure out, oh, these are beta sheets with a 4.8 angstrom spacing between the arrows and a 10 angstrom spacing between the sheets. These are the kind of clues and the ways you can use X-ray diffraction data to determine structure. And I like this because it shows it really clearly. This is usually under the hood, so to speak. And they even talked about how they did it. If you look at this, basically the problem, it was hard to prepare milligram scale structurally homogeneous fibril samples with the 42 residues. And they actually did it that showed it with crystallography. Another group did it with solid state NMR. So they can work together. It's like they put aside their differences and their rivalries, and they both solved the structure, and they got the same essential structure. To solve the problem, they basically grew their fibrils in aqueous solutions with different combinations of pH, temperature, and ionic strength. All that means is they tried a bunch of stuff until they got good fibers, okay? And look what they varied. It wasn't rocket science. You just try really hard. And eventually they got good conditions. Once you have the good crystals or the good fibers, you're able to solve these problems. And so these are the two papers that come, and I want to point out this is the A beta 1 to 42 fragment. This is an example of the NMR structure. The NMR, actually, you can see the cross peaks, and this tells them some protons that are close to some other protons. You don't have to read it beyond that. I just wanted to show you what we talked about exists in real life. But then from that, they got distance restraints. If you look at this figure, it says it's the distance restraints. They found out that this residue was close to that residue, this residue was close to that residue, and you eventually had a dimer of chains with these distance restraints, and it said that it had this structure. That's the only structure that would fulfill those restraints. So then they saw, because it's two-dimensional, the thing to realize is it forms this in 2 dimensions, and then you can stack it up because the beta arrows are pointed this way. That means they have hydrogen bonds up and down. That means you can stack this indefinitely, and that's how the fibril forms. So this is the actual picture of the atomic structure of the fibril. If you do it by charges and hydrophobicity, you see there's a lot of hydrophobicity on the middle. There's a lot of charges on the outside. The structure makes chemical sense. So they show this to you so that you can look at it and you can agree. You can look at it now and you can say, oh, that could be it. That works. That puts all the pieces in the right place. And then there's another paper that did it by cryo-electrons. So no technique is left out. We got all of those, and they saw certain spacings. They saw a 9.7 spacing, a 4.7 spacing. Same kind of thing as X-ray crystal structures. Same idea, repeated beta structure. And this, I believe this is from the crystallography paper because I've got to stay true to my roots. But this is the thing that I showed you before. This is what you modeled in class. So you can find yourself on this, and you can see where you fit in this sort of curvy shape. But this is how the fibril forms. If we can block this, we can stop fibril formation and potentially stop Alzheimer's. Now remember, there's a 40 residue fragment too, and one of the reasons why Alzheimer's is such a pain is because the 40 residue fragment, you just cut off the last 2 last 2 residues, okay? So the last 2 people in class, if they weren't on the chain, that wouldn't have really changed the way that we were modeling the structure, right? It turns out that in real life, you end up with, what we did is we did sort of the orange chain right here. In real life, if you cut off the last 2 residues, you end up with this totally different thing. The K28 person is now pointed in a completely different way. So do we have K28 here? Or F19, okay, so you're roughly pointed the same way, but you'd have different people across from you. We could actually do this, but we don't have enough time as it is, right? But the idea is, this is really complex, and who would have predicted that you can't predict these stable conformations without experimental data? But once you have the structure, you can start to design things that mess with it, because it is built on beta strands coming together, right? So some people have already been working on this. This is actually for the tau protein. It's a different protein, different fibril. But they have designed a little inhibitor peptide that will form its own little beta structure, strong enough to bind, but weak enough to keep the fibril from forming, from stabilizing. This is exactly how they worked out to make it go in. And it actually is just 4 residues that they, and it spells out something. It spells out either wink or mink, right? It turns out that this fits between, and in their laboratory tests, it prevents fibrils from forming. Now, how are you going to get into the brain? How are you going to know when to get it into somebody before the fibrils form? Lots of issues with that. The main thing I want to show you about this, fibrils will form from pretty much anything if you let them. This is what happens if you just take backbone and no side chains and run it through a computer and let it float around. For a while, it looks like nothing is happening. Then all of a sudden, you start to get structure. And then it starts to aggregate and form more structure. Look at that structure very closely. It is a beta sheet that's forming in a fibril conformation. There's no side chains holding that together. The only good news is there's no side chains. Side chains probably get in the way of this happening in many cases. But if you just have a backbone, even an all-alanine has the potential to form amyloid fibrils. And that's probably why it happens so much. The potential is there. I just want to show you a few other cases of the clinical relevance. This is from the Parkinson's causing agent, which is alpha-synuclein. And I want you to see the same thing again. You have repeated beta structure. In this case, you have a lot of glycines, alanines, valines, a lot of small residues that fit together and they form a more compact structure with different spacings. If you look down here, there's different spacings. That's just what they find, and they piece it together. And you notice the similarities and differences to what Alzheimer's has. The thing about this is this actually will fit together in both directions because the side chains are so small. You can have a repeated structure in A, it shows you how you can have repeated structure in another direction. And so you just have repeated structure all over the place in this case. Okay. So notice the spacings right there. That's what they found experimentally. And then they figured out that this model could fit those spacings. Here's another one, Staphylococcus aureus. But I want you to tell me what's different about this one. This forms a amyloid-like fibril, so it's a repeating structure. It goes up and down, looks like the other one, but do you see the really weird thing right here? It's alpha helix. So somehow it's even able to happen with alpha helical structure under the right or wrong conditions. I guess under this case, probably Staphylococcus aureus wants to kill the other cells. So from its perspective, it wants to make a amyloid fibril. So weird evolutionary pressures in this case, but you have hydrophilic is blue, hydrophobic is red. Notice how you just have helices that have a hydrophobic side and a hydrophilic side, and it builds out a fibril based from that. This is another view of it. You can see how the side chains fit together here, and on top they show you their cross alpha fibril, and they compare it to a regular beta fibril on the bottom. Notice how it's the same kind of thing where you have repeating structure, side chains fit together. Not that hard to do. And that's what makes this sort of the Halloween lecture. It's kind of scary, you know, because all proteins can go after you. This is actually a prion version of amyloid, but you see it has the same characteristics that we talked about. So this is just something that's comparing all three of them. 3 very different cases, 3 different amyloids. 2 of them are beta structure. 1 of them is alpha structure. They're all bad, okay? They all will form solid inclusions that will mess up life. But this is also a question. Is it really the solid inclusions that mess up life, or is it some structure intermediate on the path to the solid inclusions that mess up life? And this is the thing I'm really excited to tell you about, because this is fresh, new, from just published this year. Oh, this is the one thing before that. This is showing you how it's everywhere. This is just published this year, showing you how they even found it in cataracts. So remember cataracts, high concentration protein solution? They even found amyloid in those. Basically, it can happen anywhere. we have a high concentration of proteins even in the lens of the eye. Now here's the light at the end of the tunnel though, okay? So the one thing that you can see, this is a thing, and you can look at this more in more detail, but I want to give you the summary of this. This is the possible clinical therapies for Alzheimer's, for getting rid of the fibrils. The good news is you don't have to get rid of the fibrils. There are people that have lots of fibrils. These are the 8 nanometer fibrils, and right here, insoluble 8 nanometer amyloid fibrils that compromise the plaques. There are patients that have tons of plaques, but they don't have Alzheimer's. They looked at what those patients make different. They don't have as much of the 4 nanometer filaments that are on their way to being the 8 nanometer plaques. They don't have as much of a stable intermediate. So once it forms the plaques, it's actually, that's not the problem. The problem is the nanometers, is the intermediate on the way to it, which has that same structure that you formed. It's just that it hasn't stacked up as much. That's actually really good news, because that means we can go after the 4 nanometer fibrils, and we don't have to worry about the 8 nanometer, nanometer stable ones. And so they've developed antibodies against these. And it turns out that those antibodies are really promising. And I think that this might hopefully start to make inroads against it. These are the details on why I say the antibodies are useful. It goes together with a good imaging technique, so you can image how much of the 4 nanometer fibril they have. It's called PET amyloid, so you can measure how much of the amyloid burden a patient has while they're, before you'd have to look at the brain itself. This allows you to look into the brain and see the forming fibrils. And the antibody will reduce it. The antibody will bind to it, and that will allow the body to grab onto it and pull it out of the brain. So the main thing about this, if you look over here, there's some kind of things about this. So it's not going after the endpoint, the misfolded protein, but the partially misfolded intermediate. Hopefully this will, I've seen a lot of these things like go both ways, but I think that this will work out in the end. The last thing that this shows is the antibody is acting as a chaperone to the misfolded intermediate. In this case, the antibody is binding the 4 nanomolar intermediate, holding it in solution so the body comes along and clears out both of them. The antibody is keeping the 8 nanometer fibril from forming. And so by binding the intermediate, it serves as what's called a chaperone. That's the last word for today. Remember that we can have proteins that are prions that can cause misfolding of other proteins. Chaperones are other proteins like this antibody that are designed to bind misfolded proteins and keep them away from other misfolded proteins. All you have to do is get rid of the intermediate. You don't get the misfolded fibril. There are 2 kinds to talk about. Heat shock proteins is one, chaperones is the other one. These are the ones that work in the body, and the antibody that we're talking about, the therapeutic antibody, mimics the action of a heat shock protein. So heat shock proteins, they're actually pretty simple. They're just normal sized proteins, and what they do is they have hydrophobic patches. And they go around, and their hydrophobic patches will stick onto exposed hydrophobic regions of other proteins. But the good news is, if you're sticking to it, if you have like a new peptide that hasn't had time to fold completely yet, and it has part of its beta structure hanging out, this will actually bind to that hydrophobic beta structure, so it won't be able to form a fibril. So a heat shock protein, and by the way, they show up when the cell is heated. It's shocked by having heat, and that's why they're called heat shock proteins. It helps the cell deal with that. The other kind is kind of cooler and more complex. It's called chaperones. Those are huge protein assemblies, and they're essentially a box made out of protein, and they have a timed lid on top that goes on, a timer goes, and then it comes off. This is the GRO-ES, GRO-EL complex. There's a lot of scientists who work on chaperones and chaperones. Obviously, if you can make an antibody that essentially acts like a chaperone and it can potentially cure Alzheimer's, it's going to be a big area to look at. This is just a little cartoon by one of them. This is, I forget, this is one of the scientists that I think it's Susan Lindquist is the scientist who made this. But it's basically, in vitro, you can have a protein that'll be just fine in the tube in glass. But in the real life of the cell, it's a jungle in there. There's so much going on. And the newly made protein is saying, where do I go? When do I fold? What am I? Can't you recognize me? And then you have the nannies for the newly made chain that will actually be like Mary Poppins, and they'll chaperone it so that it doesn't get mixed up with other aggregate-prone proteins. And all you have to do is bind it and keep it away from the other stuff. This is an example of a heat shock protein. So this is a small chaperone. Both of them are chaperones. Chaperonins are large. Chaperones refers to both of them. But this is a heat shock protein, maltose binding protein. It binds to the exposed core. The yellow is the chaperone, and the blue is the chaperoned protein. Okay? So you can see how this definitely serves as, it replaces the hydrophobic core even, and it keeps this protein from sticking to other things. It also helps it cross a membrane. Here's another structure of a heat shock protein 90. It's in orange, and you see again, it's binding a 2-protein complex in teal and blue, and it's just binding the exposed hydrophobic core. If it's stuck to the chaperone, it's not going to stick to another protein. And that's the whole idea. These are not that complex beyond just having a hydrophobic interaction. So the main thing is, what you're doing is you're binding folding intermediates. So like if you have the native protein needs to form from the unfolded protein, has to go through a folding intermediate. And what the chaperones do is they bind this so that it can eventually go to that before it goes up. They try to keep the concentration of folding intermediates by themselves, sticky ones. They try to keep that concentration low. And so what they go after is they go after this state, and eventually they let go, and then the native protein is by itself and it's able to fold without aggregating with others. It's really just keeping stuff away, sticky stuff apart. This is the chaperonin. This is GRO-ES, GRO-EL complex. And the main thing I want to show, it keeps proteins apart by giving them their own room, okay? It actually makes a little box of proteins, and another regular-sized protein can fit inside that box. The lid goes on the box. That's GRO-ES, the blue part here. It sits on the box, and then what it does is it waits until 7 ATP are burned. The reason for that is not to use their energy, it's just to act as a timer. Because it takes about 10 seconds to burn 7 ATP, and that's enough time for the protein to get its act together and to be able to actually, it's more compressed, and it's able to find the good interactions, form the native state, then the lid pops off and the protein goes out, and it's no longer got sticky unfolded intermediates. By the way, notice that this has both kinds of chaperones. First, it has a heat shock protein, small chaperone, and then it has the large chaperonin that is the GRO-EL complex. I think it's cool to use ATP like a timer. You can imagine it like a little kitchen timer, and it counts down from 7, and then the lid pops off. But all it is a box, and this shows what it does to the protein funnel. If you have that box, the protein is going to be constricted, and so it's not going to be as spread out. Its entropy will be lowered. So its folding funnel will be much narrower. And think about it. If you have a folding funnel that's narrower, the intermediates and the kinetic traps down here are also narrower and smaller. So it's more likely to be able to find the native structure because it can't spread out as much. It's just going to be able to be sort of not quite pressed together, but sort of kept away from everything else and just allowed to come to itself. Sometimes you need a time like that where you need to just go off and be by yourself in your room. This gives the protein a chance to do that so it doesn't stick to other things and aggregate. But of course, everything has a double-edged sword in biochemistry. Some cancers actually express more chaperones because a cancer cell is stressed. the chaperones help the cell navigate, be more stressed without falling apart. And so cancer cells will make more chaperones to try to keep themselves from falling apart so that they can continue to be cancer cells. But the good side to the bad side to the good side is that now we can go after the chaperones in cancer cells. Maybe we can stop them from working, and maybe that would be a way to kill cancer cells. So you can go back and forth on that, but chaperones in general are a good thing, just not for cancer. All right, thank you for staying. Thank you for listening to How Protein Folding Can Save Your Life. Yeah, so doctors, maybe, or even protein chemists can also learn stuff like that. And I will see you on Wednesday or before.